We all know our immune system changes as we get older, right? But for years, scientists couldn't really agree on how. Study after study seemed to come up with conflicting results, leaving the whole puzzle of immune aging, well, unsolved. But now, some really groundbreaking research has started to connect the dots by tackling this problem in a completely new way. So, what was the central mystery here? It all boils down to this. How could one major study point in one direction while another pointed somewhere completely different? You can imagine, this contradiction was a huge roadblock for anyone trying to understand how our bodies defend themselves as we age. And here's the twist. It turns out the problem wasn't necessarily bad science, not at all. The real issue was that different research groups were collecting and labeling their data using their own unique methods. And that made it almost impossible to compare their findings directly. Think about it like trying to solve a puzzle, where every single piece comes from a different puzzle box, right? One study defines an immune cell this way. Another one uses totally different genetic markers to identify it. And the result? You end up comparing apples to oranges, and the big picture of how we age just stays blurry. In scientific terms, this problem has a name. It's called a batch effect, and it's a huge headache. It's kind of like trying to stitch together one giant panoramic photo from pictures taken with 10 different cameras on 10 different days with totally different lighting. All that technical noise from each photo, each batch, hides the real, true image you're trying to see. So, how in the world do you solve a problem like that? Well, a team of researchers came up with a pretty radical solution. Stop trying to compare all those separate photos. Instead, digitally stitch them all together into one massive, unified map. And this was the breakthrough. And when I say massive, I really mean it. They took data from 7 different aging studies from over 100 different people, and they integrated the gene expression profiles of, get this, over 1 million individual immune cells. Nothing on this scale had ever even been attempted for the aging immune system before. So here's how they did it. It was incredibly methodical. First, they collected all that raw data. Then they filtered out any low-quality cells or errors. And then came the magic step. They used this really powerful computational tool to digitally erase all those batch effects, all that noise. What was left was a single cohesive atlas, ready for some real analysis. And the result of this process was, honestly, stunning. Before, the data was a jumbled mess where cells would group together based on which study they came from. But after they ran their correction, the cells started clustering by their true biological type. The noise was gone. And for the very first time, researchers could actually see the real patterns of aging. Okay, so now we have our clean, unified map. The real detective work can finally begin. The team started hunting for changes that showed up consistently across all the data sets. The reliable fingerprints of an aging immune system. And the first major clue was undeniable. Across every single data set, older people showed a really significant drop in what are called CD8+ naive T cells. You can think of these as the fresh, inexperienced soldiers of your immune army. They're ready and waiting to fight brand new infections they've never seen before. And boom, another clue appeared right alongside it. Mite cells, which are a special kind of T cell that guards our internal surfaces, like our gut and lungs, they also showed that same consistent drop. Together, these two changes were some of the clearest, most reliable signals in the entire atlas. The atlas backed up other changes too. Things like an increase in non-classical monocytes and a drop in another type of naive cell, the CD4+. The picture was really starting to become clear. As we age, some of our immune populations shrink way down while others expand. But, every good detective story has a red herring, a clue that seems super important at first but ends up leading you down the completely wrong path. In this case, that red herring was a group of cells that had high levels of a gene called MALAT1. So previous research had suggested that these MALAT1 high cells might be a sign of something called T cell exhaustion, which is a key feature of immune aging where our T cells just get worn out from fighting for so long. So were these the bad guys everyone had been looking for? But the atlas revealed something really fascinating. When the researchers zoomed in, this so-called MALAT1 high group wasn't one thing at all. It was a chaotic mix of totally different cells. Some looked naive, some looked like experienced memory cells, some weren't even T cells.
It was not the consistent group of exhausted cells that everyone expected to find.
And the researchers' own conclusion says it all. They basically said that these cells showed signs of stress and damage, suggesting that high MALAT1 levels might just be a technical glitch, a sign that the cell itself is poor quality, not a true biological signal of aging. So, case closed on that suspect.
Okay, so with that red herring dismissed, the investigation turned back to the most solid piece of evidence. And this led them to the real smoking gun of immune aging that the Atlas had uncovered.
It turns out the most important discovery wasn't just that the CD8 plus naive T cells were declining. No. The real secret was hidden inside the cells that remained. The researchers decided to look at the gene activity within these specific cells in older people.
And here is what they found. Even in these so-called naive T cells, these fresh-faced rookies, there was a consistent uptick in the activity of genes tied to inflammation and cell killing. Genes like S100A4, which is a trigger for inflammation, and PRF1, which makes a protein for killing other cells, they were way more active. It's like these rookie cells were already primed and ready for a fight.
This discovery gives us a powerful molecular basis for a concept you might have heard of called inflammaging. That's the chronic low-grade inflammation that tends to develop as we get older. And it's not just some theory. It's a major risk factor for a ton of chronic diseases. This atlas shows us one of the potential sources, hiding right inside our own naive immune cells. By creating this incredible unified map, scientists have finally moved from comparing apples and oranges to actually reading a detailed story that's written in our very own genes. And that leaves us with a really fascinating question. If this atlas lets us read the story of our immune aging so clearly, what might it teach us about how to rewrite the future?